Dr. Elemento received his bachelor's degree and 2 master's degree in mechanical engineering and intelligent systems, and a PhD in computational biology, all of those in France. He completed his postdoctoral training at the Louis Sigler Institute for Integrative Genomics at Princeton, and then joined Weill Cornell Medicine's faculty in 2008. He's been recipient of several awards, including the NSF Career Award, Herschel Trust Cancer Career Scientist Award, and the Walter B. Wriston Award. He is, Dr. Elemento is a world-renowned computational biologist, has published over 150 scientific papers in genomics, epigenomics, and computational biology. Currently has 2 R01s, U24, and many other grants that supports all of those great papers. He led the development of the first New York State approved whole exome sequencing test for oncology, and has developed numerous methods, some of you may know, for assessing tumor driving pathways using computational biology. Similar to our institute, under his leadership, the Englander Institute for Precision Medicine's primary mission is to uncover roots of disease using genomic sequencing, informatics, and other technologies, and aims to empower other Weill Cornell Medicine investigators to expand their precision medicine research focus, kind of spot on with what we're doing. Much of his research, I think, would be an interest to many on the call, and his research is kind of far-reaching. One of his recent papers actually is in Cell Reports, where he was senior author on a pan-cancer analysis of how age is associated with molecular patterns and phenotypes. And actually, we did this actually in our journal club for our lab actually just recently, because that relates to some of our work. But today, he could speak on many things, but I asked him to speak about organoids, because this is a very fast-moving area. Many of the people on the line are interested in organoid medicine, including our institute. I put, as you see on my Zoom background today, I took one of the images from Daniel Brown, who's creating organoids in the institute, our Institute for Precision Medicine. Despite the relatively short period of organoid research, if you take, say, Claver's as his seminal paper as a kind of a time point, Dr. Elemento has published 12 papers on organoids recently, 1 in JCI, 2 in Nature Communications, a Nature paper on endothelial cells in organoids, and a recent cancer discovery paper. And he's now going to tell us about towards organoid-driven cancer precision medicine. If you want to ask questions, you can put them in the chat, or we will open up for questions at the end so you can ask them personally. So Olivier, it's fantastic to have you speak this afternoon. Thanks.
Well, thank you so much, Adrian, for having me. I really have been looking forward to this. Like you said, we seem to have a lot of things in common, a lot of mutually relevant interest. Our institutes actually even have the same name, so it's like clearly we're meant to work together, or to do something together, to collaborate. And I'm really hoping that what's going to come out of this talk here is hopefully ideas that may be substrate for collaborations. We're going to have sort of more meetings, but what I want to do today is really to give you an overview of the things that we are doing. I'm not going to go into a lot of detail about each individual topic. It's just kind of more sort of to give you a sense of the different aspects of the project that the projects focus on organoids. So this is the goal of my talk, is to give you a broad overview. And again, and I'm really hoping that we'll follow up with specific questions, discussions, and maybe hopefully, as I said, some collaborations. Let me just get started then. So these are some of my disclosures. Just want to, kind of highlight the fact that I may have some level of conflict regarding some of what I'm going to talk about today. Some organoids, in fact, many organoids are being licensed to a variety of different companies. We also are getting funded through industry for some of the organoid work as well, for example, with Eli Lilly. So keep that in mind. There's a bit of sort of conflict, but nothing really at the end of the day is sort of problematic. As I said, there's going to be a lot of different topics, and I want to really give the credit to the people who are really doing the work in the trenches here at Cornell, or not only in the trenches, really managing the work as well. Dr. Martin is the director of the Organoid platform. She's assisted in the direction of the platform by Jenna Moyer, who's a wonderful research technician, specialist who's really also helping in many different ways. We have several PhD postdocs working on different components of this platform, from the co-culture assays I'll tell you about, from the Crispr screens that I'm going to tell you about, the high-throughput screening component I'm going to tell you about, as well as some of the additional innovation aspect, like co-culture of fibroblast and tumor cells. So just keep that in mind. There's a lot of people involved in this whole process. In general, our institute has a lot of people involved. We are a very sort of collaborative and sort of a hub. We call ourselves a translational hub at Cornell. We are involved in many collaborations with different faculty members, physicians, physician scientists, and you'll see that these collaborations will transpire throughout the talk, I hope. So here is the content of the talk. I'm going to start with a very quick overview of our institute. It's called the Institute for Precision Medicine. We have an E that stands for Englander Institute. We got some essentially endowment for the institute, and I think that's helped us tremendously in terms of funding many of the programs I'm going to talk about today. So I'm going to give you an overview of the mission. I want to give you a sense of our vision about using organoids for precision medicine. I'll tell you about the platform itself, how we grow organoids for all kinds of different tumor types. I want to tell you briefly about the high throughput screening, drug screening platform that we've set up. I think it's a cutting edge type of system. I'm very proud of it. It's taken a long time to set it up, but now it's really generating a tremendous amount of data. And I think this is going to be a game changer for us in the future to have the ability to do this kind of screening in-house. But as I'm sure you all know, we are just really at the beginning of being able to create organoids that are good copies, good reflection of the patient's tumors. And we need to work towards building better organoids. And I'll mention some of our efforts in that space. And I'll try to finish with thoughts and maybe ideas and our vision for the future regarding establishment of a clinical utility of organoids, because I think this is a very important topic and a topic that has not yet sort of been fully established, and something that we need to collectively sort of work on. So I like to start my talks with a definition of what I think precision medicine is about. As I said, we are the Institute for Precision Medicine, just like you. So my view of precision medicine is that essentially it's a form of personalized medicine that is enhanced by technologies. We have technologies now such as high-throughput sequencing that allows us to sequence somebody's genome in 48 hours or less. We can actually sequence lots of genomes in just a small amount of time. So that's an amazing technology for precision medicine that's helping us in so many different projects. We also have analytical tools such as AI and deep neural networks is just one example of such tools that allow us to integrate data at multiple layers, extract more information, predictive information, for example, from images, which also is really important when it comes to personalizing medicine more than it's been in the past. So it's really the core of precision medicine, all these technologies that are also going to be the core of the talk today. This is an overview here of the cancer precision medicine program at Cornell. This has been something that we've set up for several years now. It's traditionally been focused on sequencing, especially sequencing of DNA. So we actively do biopsies or get surgical samples from patients with cancer, especially advanced cancers, and we sequence the DNA and sequence normal DNA as well to serve as a control, to be able to produce reports that we discuss in case conferences and that contain or lead to highly personalized treatment recommendations. We've actually sequenced now more than 2,000 patients. We are still very active when it comes to this approach for personalizing medicine for cancer patients. We also do RNA analysis, which complement the DNA analysis in very nice ways. This is how we report information back to patients. We have these very nice reports focused on sort of summarizing information from whole exome sequencing. And again, this has led to lots of recommendations for treatment, lots of really improved outcomes across the board for many different types of cancer patients. in a different talk, I would continue on this theme and tell you much more about this aspect of our precision medicine program. But really, this talk here is focused on kind of an orthogonal, or let's say a parallel approach for precision medicine, which is using organoids as a way to drive the precision medicine process. And it kind of, starts with recognizing that as much as we, would like to, as much as we made a lot of progress when it comes to reading people's genomes, whether it's germline or cancer genomes, there are still so many cancer mutations that we don't understand, that don't have a clear function, that we can't act on. We can't essentially match patients to drugs or treatments just because we don't understand what these mutations are doing. So that's obviously led to some frustration that we are trying to address using a different sort of approach. In fact, we've actually looked at this in detail. We published several reviews on the topic, but it's probably in the order of about, depending on where you do it, about 25% of patients on average that benefit from a genome-guided sort of approach to precision medicine. And so that really leaves a big delta of patients who are not served by these kind of sequencing sort of type of assays, and therefore, again, unmet needs that we need to address one way or another. So, it started with a question, right? I mean, just like probably your efforts, but, one idea would be, one crazy idea would be that maybe, it's possible to, not just try to read the genome or understand directly what's happening within patient samples, but maybe just empirically test all kinds of different compounds on samples coming directly from patients to be able to essentially test a whole spectrum of therapies and maybe find the ones that are most effective for each individual patient. This is kind of a dream in some ways, and this is something that we continue to be excited about, and that we're trying to essentially make a reality at Cornell using this effort. As you'll see, there's a lot of dimension to it, but this is really the guiding theme of this whole approach. And this is a great paper that I've tweeted about. Really a wonderful review by Dr. Welm at the Huntsman Cancer Institute that really sort of, in my view, I think is summarizing the field in a very nice way, and really showing all the potential that exists with this idea of deriving cultures from patients and being able to test different hypotheses on these tumors grown ex vivo. And what's exciting to me is especially this idea of novel combination, because I think that the genome, I think, is especially limited when it comes to coming up with combinations of therapies that may be quite effective for each individual patient. And I think that it's going to be hard to establish, a framework where this is, we can guess what combinations we should use. I think this is where I think the organoid sort of screening and could really have tremendous impact. So this is how we started a few years ago. This is a work that was done by Mark Rubin here, led by Mark Rubin here at Cornell when he was here. Shanta Poli was, as you see on the right side here, really led this work. We started essentially a very systematic effort to grow tumor organoids using the Hans Cleaver sort of approach, more or less, but across many different tumor types. So our approach is really not focused on one tumor type. I think just like yours, it is a pan-cancer sort of type of approach for growing organoids. And I think this is, again, I think what is making us, just like you, I think, reasonably unique in that space. We're not focused on one type of cancer, like pancreatic cancer. We're just doing this across the board. But we started this a few years ago. We published several papers about this already. And part of the sort of some of the decisions that had to be made early was how to exactly do this kind of in vitro sort of type of analysis. Obviously, there's a lot of interest in kind of going beyond in vitro and using, for example, PDX models, which we like to use. But I think we and others have realized that the time it takes for PDX to be established is just not really compatible with a kind of a treatment goal for a functional sort of precision medicine sort of type of approach. Likewise, you can say, well, maybe cell lines, could potentially also work. And I think cell lines, have been very helpful in the past when it comes to, again, precision medicine. They don't recapitulate, I think, the clear heterogeneity that you see in patients. They adapt, to a different sort of way of growing. probably evolve, much faster than we would like them to do, and likewise are not, ideal when it comes to sort of reproducing the features of tumors ex vivo. So that's really led to our focus on organoids, which is in the middle. It doesn't take too much time to grow. We can do high throughput screening or semi-high throughput screening, and we can model, as you'll see, tumor heterogeneity using the system. It's obviously one of the limitations is the fact that it doesn't always capture the microenvironment. But again, this is something that we're actively working on, as you'll see in the next few slides. And so this is sort of the approach that we're taking. This is our precision medicine sort of type of approach. Starts with a protocol that we have to enroll patients, where we can get tissue from those patients. We work from fresh tissue, of course. We have universal consent. We are we have a team that's ready to intervene when a patient is enrolled and when tissue is made available. We can capture the tissue and essentially process the tissue right away. We have a whole team that's sort of focused on doing this. There's a lot of additional steps, as you know, in terms of making sure that what we grow is what we expect, what we want to grow. From pathology review to sequencing, we're actually putting a tremendous amount of effort into making sure that we understand exactly what we are growing. And I think that's also something that's very important. There's a quality control component that is critical in this effort. Then we actually are doing in-house drug screens. We started with doing this for a company in Seattle, but we decided that we needed to have the flexibility to choose a lot of different parameters, try different things, which led to us investing in a very exciting platform that I'm hoping to be able to show you in the next few slides. So one of the things that's also obviously important when it comes to sort of organoids is that we want to use each one of them for a precision medicine type of approach. But every time we are successful in establishing a new model, we also can freeze those models and essentially maintain a biobank of organoids that is growing and growing. Right now, this is a number that I just got today. We have 213 different tumor organoids in our biobank. And as you can see here, they're really across the board. Obviously, there's more colorectal cancer than other tumor types just because of the fact that the success rates are higher in colon cancer. But as you can see, we've been able to grow organoids for all kinds of different tumor types, including tumor types that are pretty rare. And I'll maybe also give you a little bit more insight into this later. These are examples of these organoids from bladder tumors or bladder organoids or stomach cancers, kidney cancer, melanomas. Melanoma organoids, as some of you may know, is actually not so easy to grow. There's been some work in the field, but probably not as much as there could be. One of the things that we've observed, and that really I think justifies even further in some ways the concept of organoids in melanoma, is that when we grow melanoma organoids in 3D in Matrigel, we actually show that we actually maintain the pigmentation of the organoids. This dark pigmentation, it goes away when you grow these organoids or these tumor cells in 2 dimensions. So it really kind of reminds you that there is something special about growing these tumor cells in 3 dimensions that essentially keeps them in contact, keeps the crosstalks between cells that you see in tumors intact, and clearly is preserving some fundamental physiological properties of these tumors. And so we have actually a follow-up on this, actually including some ideas about why this pigmentation is maintained, but this is for another day. So as I said, we do extensive QC on the organoids. We do pathology analysis. We have pathologists who look at the H&Es from the cultures and make sure that what we grow essentially looks morphologically like the tumors that these cultures are coming from. We do this. The proof of concept is being done both in organoids but also in PDX models. And likewise, we've been able to show across different types of tumors. This is part of a 2017 paper, the first paper that we published to really demonstrate the breadth of application of organoids. The sort of in vivo models and ex vivo models, do behave in the same way morphologically. We maintain the same sort of overall features. So I'm just done with that. Okay. As I said, we do a lot of genomic sequencing, and likewise, this is also for us a way to assess quality control, making sure that tumors established ex vivo don't evolve too much. We maintain the same kind of genomic information as the tumors that you see in the patients. That's an example across different passages and going from organoids to PDX. The sort of overall genomic sort of maintenance of these cultures. Sorry, when I move, there's a little glitch here, but hopefully okay. So that all being said, so that paper, was published in 2017. We actually have done so much more sequencing now, and we see that, unfortunately, concordance between organoids and primary tumors is not always as high as we would like. That's, again, I think that's part of the sort of the effort. But we do see that typically what you see very often is that the driver mutations tend to be maintained between the primary tumors and the cultures. The passenger mutations may be lost, but a very significant fraction of the driver mutations tend to be maintained, both when we look at copy number alterations as well as point mutations in indels. And we also do this using RNA sequencing, which also kind of provide us with another readout of the extent to which organoids, the cultures sort of cluster with the original primary tumors and clusters among themselves for the same tumor type. One of the sort of fundamental questions that the field is really now trying to address in general, whether it's primary tumors or organoids, is the extent of what we call plasticity within tumors. There's different ways to think about this, but I think one thing that's emerged, and we published on the topics now multiple times, others have published on the same topic in other tumor types. But what you see very often in primary tumors now is that each tumor, in the past was classified as maybe belonging to one subtype. For example, basal or luminal sort of subtype for some tumors. What we've seen, and with the advent of doing a lot of single-cell RNA-seq, is that in fact every tumor is actually a collection of different subtypes. Different subpopulations that have essentially different that essentially belong to different subtypes. Potentially, sometimes with the same kind of genetic underlying sort of profile. Which is sort of one way to describe this is plasticity. Essentially, there is ability with the same genetics to kind of transition between different subtypes. That's important because that's what we see in primary tumors. And I think one of the key questions is really whether we also see the same thing in organoids. That's a question that we've tried to address, at least in some tumor types. This is a study that we published in 2020. On brain organoids, where we did single-cell RNA-seq of brain organoids, and compared with essentially compared the diversity of cell populations that you could see within each organoid compared to primary tumors. And one thing that we saw is that we actually saw a pretty good amount of sort of maintenance of plasticity, essentially the presence of the same different cell types within each individual tumor that we see in the patient's sort of tumors, directly. And there are different types of cultures that you can do ex vivo, but the glyco model, which was really the theme of that particular sort of paper here, which is essentially a brain tumor organoid model where you culture glioblastoma sort of tumors within the context of essentially normal cells, like almost like a mini brain that you use as the sort of the context in which you see glioblastoma stem cells and you grow a glioblastoma. That type of approach was able to maintain the plasticity that you see in primary tumors really nicely. So I think it needs to be kind of extended to other sort of types of organoids, but I think at least it is a good sign, I think, in general, for ability of organoids to maintain the plasticity that you see in primary tumors. This is a slide here just to show you in one slide and one very quickly that we've done this for a long time now, and we've been recognized in lots of different ways. One of them is we've been invited to join the Human Cancer Model Initiative, which is a network of sort of sites that produce tumor organoids and put them in common into a very large database stored at ATCC, and we are one of the production centers. We're producing organoids and sharing them with the network so that we can make these models available to everyone in the scientific community. Shifting gear here a little bit, this is work that we did also a little while ago. As I said, this is some of the early high throughput screening results that we did with a company in Seattle. This was some of the organoids that we sent for screening. This initial sort of type of screening was able to screen in the order of about 100, 150 drugs at the same time. These are some of the results here. This is just 2 patients with colon cancer with slightly different genetics, as you can see here. But showing, I think, something that's really important and really justifies organoids in general, which is that essentially those 2 organoids responded to completely different drugs and responded to drugs that they were not connected to the genetics, at least to the mutations that we found in those patients. So again, I think early proof of concept of high throughput screening. And I think, justification of the model. We also in the paper, and that's something that, we still haven't sort of fully reproduced yet in-house, but as I said, tremendous potential for screening for drug combinations with organoid sort of type of models. We did that actually in that particular paper and showed that there are certain drugs, single agents that are completely ineffective. For example, celecoxib in this particular patients at the top here. But if you combine it with trametinib, which is by itself effective, the combination is actually tremendously effective. And so I think it's a great example of screening for combinations that maybe are not obvious given the genetics of the tumors. As I said, there was a tremendous amount of interest in being able to do our own screens in-house. We joined forces with multiple groups, here at Cornell, the Cancer Center, Dr. Fine, who focuses on organoids in the brain. And we essentially bought a customized robotic system to be able to do our own screening for drugs. So this is a great platform, really exciting stuff. I'm just going to show you a little bit of, how it works. It takes this is kind of a long movie. I'm just kind of showing you a little excerpt, for you to get a sense of the system itself at different time points. So basically, the system does everything. It does everything from being able to essentially serve as an incubator, as well as image cells, tumor cells as they are being treated. It does the pipetting. It does, essentially the whole process. And so this is how we use it. We basically do it in 2 steps. The first step is a hit assay. So we test lots and lots of different drugs at a very small number of let's say, dosage or concentration, somewhere between 1 and 3 concentrations, just to give an initial sense of the effectiveness of each individual drug. And then using the robotic system, we do a full dose response assay for the drugs that came up in the first initial screen. Again, the parameters can change. The robotics is very sort of flexible, and there's lots of parameters that can be done. This is an example of a screen results. This is from one concentration. This is a patient with esophageal cancer. This is testing close to 500 compounds, some FDA-approved drugs, mostly FDA-approved drugs, but also some investigational drugs. This is a nice opportunity, by the way. We are expanding the library because there's a lot of clinical trials happening at Cornell, or sort of not too far from here. Investigational drugs that our physicians or scientists are interested in. So we are actually trying to help our community by adding those drugs into the library so that we can generate more data also for these other groups, so that for example we can help them decide where a drug is most effective in. And just as an aside. So what you see on the left side here are the drugs that were most effective in the context of this one patient in our screen done in-house. So this is kind of the same screen, but where the drugs have been kind of clustered based on drug target. And this is even more clustering based on the type of target, kind of the pathway in which the target sort of. What you can see here, for example, is that this patient here was actually quite sensitive to the molecules that target the cell cycle, cell cycle inhibitors and so on. This is another patient here with a different type of disease, a colon cancer patient. Likewise, you can see on the left side, some drugs were actually quite effective at 1 micromolar. As I say, using the robot, we're actually doing the the drug response curve over different time points. This is taking some of the best hits from a particular sort of screen, and showing that they continue to be effective according to dose response curve, and that also allows us to calculate an IC50. That I think is important. We also include drugs that were shown in the hit screen to not be effective as controls, of course. And this is showing the results across different patients here. This is again coming from our own screen. Some really interesting things that sort of occur. For example, on the right side, top right here, what you see is about multiple patients tested with folinic acid. Most patients don't respond whatsoever, but one patient actually responded really well to folinic acid. So that's again an outlier, but some very interesting sort of observations coming out of this analysis. Part of the the beauty of that about the high throughput sequencing, the high throughput screening sort of approach is that it's actually a very sophisticated imaging sort of system as well. It actually allows us to do confocal analysis of each organoid. So we can actually quantify, tumor shrinkage. We can actually quantify the volume of each organoid. We can track hundreds of them at the same time, or even thousands of them at the same time, and really quantify lots of different things. Not only sort of viability, but also things like migration, or we can quantify all kinds of things when it comes to all the features that we could potentially want to track and modify using drugs. What we are working on now is kind of an image analysis sort of type of assay. We are trying to move away from cell type tableau, which is what's been used in all of the screens that I showed here so far. But we're trying to move towards kind of an image analysis sort of type of assay to be able to estimate cell viability based on just confocal images. And we're actually showing that we can do this. This is actually great in terms of being able to potentially do screening at different time points. Get more granularity when it comes to the information that you get from the screens. So this is just to give you a sense of some of the results. As I said, I would like to spend some time telling you a little bit more about our attempt to improve tumor organoids, because I think that's really important. We are trying to do this in all kinds of different ways. As we all know, organoids are sort of meant to reflect the complexity that you see in tumors. I mean, they don't right now, but I think this should be our goal. And the reality is that, as we know, tumors are pretty complex, right? They have all kinds of different cell types. They have some kind of structure. And I think it's known by now, or at least there's a very strong evidence that the way by which tumor cells respond to treatment and to drugs is potentially dictated, or at least influenced by the microenvironment. And so I think that it is really important, especially for some tumor types, that we be able to reproduce ex vivo some of the key features of the tumors that these cultures are coming from. I'm going to try to attempt to give you a sense of how we do this. So one of the things that we've been doing now for some time is to try to culture, or to essentially grow immune competent organoids. That's obviously the goal here is simple. We want to be able to test immune therapies on the organoids and combinations involving immune therapies and targeted therapies. That's obviously not feasible if what you're growing is just tumor cells. So we've actually embarked in a very sort of systematic project where we're getting PBMCs from patients. We are trying to expand those PBMCs. We're trying to also get tumor infiltrated lymphocytes from the tumors as well. This has actually been a little sort of complex. We learned a lot on how to do this best. Now what we do is, when we get a biopsy, we actually split the tissue into two. One goes for organoid growth, the other half goes for TILs isolation. And we tried to do both at the same time, and it just didn't work very well. And so we this is sort of what I was telling you about. We use IL-2 as a way to expand the TILs. And we do all kinds of analysis to make sure that what we expand, is obviously sort of what we expect to expand. But the goal here is, once we have T cells, expanded T cells, and tumor cells, we basically merge them together into co-cultures. And the goal here is really to use those co-cultures, for example, for the assays that I mentioned. And this is some of the additional sort of type of analysis that we do on the co-cultures to essentially try to credential these co-cultures and make sure that, again, we are growing, for example, T cells at have the right features and respond to the right sort of signals. So here's an example of how we use this. So this is showing here that we can take co-cultures, stimulate them with anti-PD-1, which is pembrolizumab, essentially PD-1 inhibitors. And we see that we are actually getting induction of different pathways. We're getting inductions of interferon gamma, which is what you see here as a result of anti-PD-1 treatment. We're getting induction of coenzyme B. We're basically essentially, really demonstrating that the T cells that we grow using this method are functional T cells and respond to stimulation in the same way that they would respond in patients. And so now we're starting to do high-throughput screening. And this is early stage, but our goal is to start to do high-throughput screening. This actually, the conversion of this kind of assay, which is very flow cytometry based, into a high-throughput screening. So the type of assay is actually more complex than it sounds. We can't do high-throughput screening and combine this with flow cytometry, as you can imagine. So we actually are working on a imaging assay to be able to quantify essentially the ability of T cells to kill tumor cells in sort of using an imaging sort of type of assay. This is what you're seeing here, using different types of markers, and UV488 caspase-3 for cell death, and cell trace for seeing the cells themselves. And as you can see here, we're seeing basically sort of cell death. We're able to quantify cell death in the context of this kind of imaging type of assay, in the context of co-cultures. And so I think this is sort of something looking quite good. We're doing co-cultures with fibroblasts as well. This is something that's just getting started, but excited also to see how co-culture with fibroblasts could potentially impact drug response. I think that there's a literature that says that it does. I think we need to establish this, in more detail. But I think this is a student in our group who's focusing her PhD on this topic. She's able to isolate fibroblasts from patient samples and show that these fibroblasts are actually expressing the right markers. What we're seeing, though, is that there's a tremendous amount of diversity among fibroblasts, cancer-associated fibroblasts. They tend to express different markers. So they are kind of patient-specific. And so, it also means that we just need to take this kind of a patient-specific approach to the fibroblast derivation, just like we do for T cells and for the tumor cells. I think that's really, really important. As Adrian, you mentioned, we also work with Shaheen Rafi here at Cornell to demonstrate that we can essentially grow organoids in the context of a vascularized sort of type of system. Shaheen is a great system to essentially grow blood vessels and demonstrate that we can seed those blood vessels with tumor cell cultures, and we can essentially recapitulate some of the interactions happening between blood vessels and endothelial cells and the tumor cell cultures, and show that indeed it also does impact some level of response to drugs in ways that we need to understand better. But I think this is a great example also on the importance of co-cultures with different types of things. One thing that we can do with those is also to test CAR T cells. We can actually flow CAR T cells in the context of these, in the context of this kind of functional vasculature, and then test additional approaches for targeting tumor cells, using this kind of system. One more thing that also is really important. All of the cultures that I've shown you so far using matrigel, and obviously matrigel, is the sort of the traditional approach for growing tumor organoids. There's a lot of issues, about matrigel and the extent to which it really reproduces some of the physiological features of matrices that you see in patients. One of the more fundamental issues is that, it's actually not available. There's actually a global shortage of matrigel. And so we're exploring actually actively how we can grow tumor organoids with our matrigel. We published this paper. This paper is actually accepted now, but where we showed that we can use essentially kind of a smart matrix sort of type of approach. It's basically a peg mouse sort of type of approach to essentially create matrices that we can use to also personalize these extracellular matrix sort of type of approaches. We're basically doing proteomics on patient samples and trying to understand what kind of matrix sort of structure you see in patients, and essentially trying to reproduce matrices that have the same kind of collagen sort of signals and peptides that you see in the primary tumors. That's allowed us in this paper here to demonstrate that we can grow certain types of organoids. This was focused on prostate cancer, but in ways that suddenly changes morphological features of tumors, and typically in ways that we think is a bit more reflective of the tumors. Is, we're able to better reproduce, for example, features related to invasion using this kind of matrix compared to matrigel. Matrigel, some of you may have seen that, but it's actually, it kind of tends to shut down features related to invasion or migration. These matrices here that are kind of more patient-specific, they seem to be better at preserving at least, some of the invasion-related features that we see in patients. We use this for lots of different things. We've actually done, sort of drug screens on some of those matrices. One of the things, this is kind of a different story, but kind of connected to the ones we've grown these prostate cancer organoids, this kind of matrix here. Very interested in novel drugs, novel treatments. And one of the treatments that came out, as part of another effort is ONC201, which we know from other sort of type of studies is very effective in the context of tumor cells that express a tremendous amount of dopamine receptors. This is a longer story here, but prostate cancer organoids grown in this particular type of matrix, just like you see in primary tumors, actually express a lot of dopamine receptor 2. And as a result, potentially actually quite sensitive to ONC201. This is actually very important because there are very few drugs that are effective in the context of metastatic prostate cancer, especially the more aggressive type that this picture is all about. And so this really paved the way for additional work focused on ONC201, this DRD2 inhibitor, which we took also to an in vivo setting and showed that it continues to be quite effective. So it's kind of an interesting story where a better sort of type of matrix led to essentially more physiological, better transcriptional profiles, more connected to what you see in the patients, and paved the way for discovery of therapies that could potentially be effective also in patients. Let me see. Just a few more slides, and then we can hopefully have a discussion. We are very interested in doing CRISPR screen on tumor organoids. We actually have a postdoc who's focusing her work on this. Really exciting stuff, actually, that as she's able to get CRISPR screen to work in organoids or cultures. We've done this in breast cancer as well as in other sort of types of tumors. This is exciting because there are lots of applications for this, as you can imagine. Discovery of novel targets in organoids, something that's really, really obviously relevant. But I also see a potential for this as a way to pre-screen organoids for certain types of therapies. The beauty of this is that in theory you can do pooled screens, right? So you can actually test all kinds of weaknesses and essentiality features in tumors using a pooled CRISPR screen. So you can test essentially a lot of hypotheses, using this kind of format, which maybe is a bit faster than doing this using high-throughput screening, or a bit sort of more scalable if you want. The downside of doing CRISPR screens in this kind of setting here is that because the tumor cells don't grow very quickly, unlike what you would have with cell lines, it's hard to get to the point where you have enough cells to be able to do a full genome-wide sort of type of screen. So we tend to not do that. We tend to do, for example, CRISPR screens using 1,000 genes or 500 genes, but still we can cover a lot of different sort of territory using this kind of approach. This is sort of some level of details on the assays, just showing here a proof of concept using one gene that we know is essential and being able to reproduce this in organoids. And this is here showing the result of sort of essentiality, essential genes detected using CRISPR screen using organoids. So clearly some interesting results that we are now trying to expand. And this is in a nutshell here again, sort of our vision about organoids. We know that there is a lot to do before we are able to have this as a clinical tool. There's a lot of sort of optimization that we need to be able to do, but I think I try to cover as much of the spectrum as possible so that you can see that we're working on every single one of these aspects. But our goal is really to be able to do this for patients reasonably soon. We're focusing now our initial attempt or initial sort of clinical development assays, if you want, or clinical development work on patients with high risk of relapse, let's say in colon cancer, for example, with Manisha here. We started working on this. We basically, as soon as patients show up, these high-risk patients who we know are very likely to relapse within 6 months, we go on organoids with the idea that even if it takes a few weeks or a few months to go organoids and do a high-throughput screen, by the time the patients relapse, we'll be ready and have additional information that we can use to treat patients using the results that we get from the organoids. So this is what we're trying to establish right now as proof of concept. In the long run, just to be clear, we love to be able to not have to do organoids. One of the things that we're hoping to learn is how to better connect mutations, the many mutations I showed you at the beginning of the talk, with response to different types of drugs. And we're kind of inspired by the example of this one patient here. This is actually a brain cancer patient where we had when we looked at the patient's tumors, we saw a P10 deletion, and we actually did the high throughput screen. We actually saw that many drugs in that patients that came up as responsive or effective in this particular organoid were actually PI3K inhibitors and AKT inhibitors, which is exactly what you would expect in the context of the P10 loss. So that's an example of how the genomics could have predicted the drug results. But we know that's not always the case. But really, our sort of ambition is to do this across many hundreds, if hopefully maybe thousands of different tumors, and essentially be able to start making connections between mutations and response to drugs. And down the line, be able to say, well, look, if you have this mutation, you're actually sure that you're going to respond to this particular drug, and you don't even need to actually do the high throughput screening. So this is kind of our dream, and that's going to take a long time. But I think our goal is essentially to put ourselves almost out of business, using this kind of approach. It's going to take a while, but this is the direction that we are trying to take here. And I think that's about it. This is my last slide here, just to maybe summarize a few kind of questions and things that we think are important, which I tried to cover. Do organoids recapitulate patient response? There's a lot of literature that shows that it does. We have some data that shows that it does, but I think we need more data. And I think this is something we're trying to generate now. There's lots of different ways to think about organoids. Whether we need to sort of one way that I haven't really discussed, for using organoids is to essentially create a library of organoids across many different samples and organoids. And when you have a new patient, you can essentially place a new patient's organoids within the context of this very large universe of organoids for which you have drug response. It's like a closest patients to the type of approach. And I think this is something that we should seriously consider. The idea here is that you may not even have to, once you have this huge collection of drug responses, you don't necessarily have to actually do the screening again. You can just place your patients in the context of this very large sort of functional universe, and I think that may actually be a good way to proceed. And that's something we also need to establish proof of concept for. Core cultures, whether they affect drug response, we have some anecdotal evidence that it does, but I think we need to study this in more detail. And the trial that we now have started actually in patients with high risk of relapse, I think is a great opportunity to do this prospectively in a way that will inform us on feasibility and potential clinical impact. Down the line, to be clear, if this is really to become a clinical assay, it can't be done in the research setting. It has to be done in essentially a CLIA setting. And the only way to do this, here at Cornell, and in fact, this is really across the board, is to collaborate with clinical labs that, where it's possible to establish this kind of workflow, this kind of complex workflow as a clinical workflow and return results back to patients. This is something that needs additional work. I'm not even sure that some of the accreditation bodies like New York State for us, are fully ready, fully sort of prepared to actually assess this kind of approach. But I think they're going to have to because I think this is really the future, and I think this is really important, and it will give hopefully so many more options to patients. I think this is why we do our work. And I will stop here, and I will be very happy to answer any questions. Thank you so much for listening. I hope I wasn't overwhelming or too much, but since we hopefully have a bit of time, very happy to answer any questions that you would have.
Fantastic, Olivier. If you want to stop sharing your screen, that was perfect. Very related to a lot of the work that people are doing here. not surprisingly, there are a lot of questions in the chat.
Very good.
And
Very good.
This is good. And a lot of them, it's a cutting edge technology that I think you well said is that, people think this could be transformative in terms of, functional selection of variants, for example, for drug testing. And so normally I would open it up for questions, but maybe I'll try and shoot through these questions so everyone can hear, your thoughts on them. Most of them are technical related to organoid development because it's still, there's still that questions about how you grow them. So I'll just run through these pretty quick. So when you talk about using TILs for the immune recombination, the TILs and the PBMCs, did you try tumor-associated macrophages? Have you tried that?
We are doing that. We actually have the same student, postdoc, who's working on the sort of TILs, actually is also doing cultures of macrophage. We have actually some success. The derivation of macrophages, as you know, is completely different. You can't actually isolate macrophages, from tumors in the same way that you isolate, TILs. You essentially have to get them from PBMCs, from essentially derivation sort of process. But we are doing co-cultures. And likewise, really eager to understand how, one, maybe we can detect or identify therapies that could potentially be, able to, let's say, for example, transform M2 macrophages into M1 macrophages, or take immunosuppressive macrophages and make them, more, immunoprotective. So I think there's a lot to be done here, but and we are starting. We actually have quite a bit of data on this, but nothing that I can present now. But certainly something I'd also love to discuss with you more if you're interested.
Similarly, a related question: can you use the organoids to look for molecules that can enhance tumor infiltration, or presumably block tumor infiltration or TIL infiltration as well?
Yeah, so I think that's an excellent question. Kind of turning cold tumors into hot tumors, I think is a big, theme, or a very important theme. With the kind of image analysis, and I think this is really, where the image analysis, the readout essentially of the screen is really important. We can do sophisticated image analysis to be able to quantify, for example, the extent to which we can mobilize immune cells and kind of make them migrate maybe deeper into organoids. We haven't done this, but this is actually an excellent question, and I think this is absolutely something that we should be doing, and I hope we'll do this in the near future. That's a very, very good point.
Two technical questions on the screening assay. How long does it those screens that you showed us, say those 500 drug whole screens, how long does that screen take, like from start to finish, like a ballpark days, for example?
So if we start with organoids that are already kind of put into wells, I think it's a couple of weeks. It depends a little bit, but that's typically in a matter of weeks, 1 week to 2 weeks. Yeah.
And these organoids are presumably growing at different rates. So how are you standardizing the time of exposure? Is it all 3 days, or are you doing that depending on the growth rate of the organoid?
I think this is an excellent point. So we actually seeing that. So we are very interested, I'm sure, just like lots of people in PARP inhibitors as an example, right? PARP inhibitors we've been seeing, tend to be effective after a bit longer of time compared to other drugs. So this is not an easy question, but I think every drug is a bit different when it comes to the amount of time that they need to be to become effective. And so we kind of need to, maybe almost, I don't know, maybe taking a kind of drug-specific sort of type of approach, for this, where the time that for which we measure things in this kind of high-throughput screening may actually be, dependent on what kind of drug, what class of drug, we're looking at. That's a very important question.
Yeah, there's an interesting question about size of organoids. We had a talk actually from Jeremy Rich, who's at our cancer center. He's the deputy director in the cancer center, who was one of the first people to do brain organoids. And he's shown that when you grow them really large, they become hypoxic, and that alters the stem cell niche. And the question actually in the chat, it's kind of related to that. What's the issue about small versus large, which presumably is an issue when you get into those screens, yeah?
I think it's an excellent question. I'll be honest with you, we have not looked at that, but I think this is a very, very important question. And I think we should be looking at this more closely, because you're right. When you put these, little sort of tissue chunks, in wells, there's all kinds of, there's a distribution of sizes, right? And so that's something we need to look into closely. One thing that I can say is that typically we don't rely on one chunk. We're tracking multiple chunks per well or per drug, so that we can take the average of multiple different sort of mini chunks, just so that we can not be too sensitive to this kind of site-specific phenomenon. But that's a very important question. We should be looking into this more.
Yeah, there was a question about how they are changing over time. You showed this one organoid where the copy number doesn't change over time, and I just wonder how much have you done that? I always show we show n=1.
Yeah, exactly.
How many don't?
Absolutely.
How many don't show that?
Yeah, I think that the reality is that they I think they change. Maybe so it depends on how you look, right? So what I showed was a copy number alteration sort of type of plot, and maybe copy numbers tend to be a bit more stable compared to point mutations. So I would say that we actually have a gigantic amount of genomics data actually now that we're in the process of analyzing with multiple time points, and I don't know that we have the full answers to this at this stage. I'm hoping we'll have those very soon. But that's a very important point as well, because the bottom line is that you need to know what you're growing. If a tumor is evolving and changing after some time, then we need to know. But the only thing is, obviously the idea of these kind of cultures, that they're kind of short-term cultures. We can freeze them, but we're never growing them or passaging them long enough that they really are. That the pressures to change are very, very significant. So the hope is that they won't change too much. But I think it's also it will depend on the cultures, on the patients, on the tumor types. Lots of factors will impact the results. We'll see.
Yeah. Okay. Interesting question. So you talked about a lot of the tumor microenvironment, all these endothelial cells, fibroblasts, immune cells. What about the tumor microbiome?
Yeah, I mean, an excellent question, right? When it comes to, for example, colon cancer, right, where we know that there is some level, well, at least there's a sense that there is some level of maintenance of maybe or at least some level of inclusion of certain types of microbes or maybe even viruses in some cases. We have no data on this, and so I think that's certainly an important topic. I just don't know the answer. We have not looked at that whatsoever. We can probably look at that in the context of maybe we have a lot of RNA-seq data, and so that seems like maybe something that maybe is in our data, but we just haven't looked at it.
Yeah, we've started to look at that in our data actually, because I think they're going to be there, and I guess the question is, can you modulate them and stuff?
Yeah, absolutely, exactly.
Yeah, a question I was going to ask actually, and someone else posted this as well. I was interested in the barriers to clinical implementation, how you just ended, because obviously all the data says, in theory, you can do this. There's enough time to grow an organoid, screen by whole exome sequencing, find the drivers, predict the drugs, give it, and then give it to the patient. Yeah. What's the barriers then to doing that? Presumably, and the question I guess in the chat is it? How are you doing that with pathology? You kind of stated this. There isn't really a way to already have regulatory approval of that. Yeah. So how are you actually actively looking to do that, or are you speculating that you're going to do that?
No, we're actually actively looking to do that. We've had a lot of discussions with pathology here to explore how feasibility. I think part of the question is, what do you need to have in pathology? Obviously, for example, just to give you an example, this gigantic robot that we have, it's like you saw from the picture, it's a big operation. I don't know that it's really feasible, honestly, to actually put that robot in a clinical lab, and at least we may not have enough space just to be very practical, for doing this. But in a sense, the robotic system is maybe one component. Being able to, demonstrate that we can, for example, take a patient samples and reliably derive cultures that always kind of look somewhat the same, where you don't get essentially a different result every time. I think this is something that, honestly, the field has not really shown. And I think we need to establish, those quality metrics. This is the essence of a clinical test. That's actually in New York State. What they ask you to show is that you do the assay multiple times on the same sort of material, and you get the same results. I'm not yet sort of sure that this is going to work as well as we'd like it to work. So maybe there is a different thing there. Maybe there is a trick, or maybe it's I don't know. We can look at sort of populations of organoids, or try to get multiple cultures and merge them into one culture so that we, sort of average out, the effect of, the sort of what you get from different cultures. So I think there's a lot to do. Once you kind of put yourself in this kind of clinical sort of mindset, there's a lot to do to really demonstrate that what we have, can be a clinical assay. One thing that is really important is that our pathology colleagues, that's what they do, right? I mean, that's for mindset, so they're kind of really pushing us to, demonstrate and provide the data and show that, there's reproducibility and, that it's feasible and, to essentially operate the whole workflow in the clinical setting. But I think it's the early days, but as I said, I think it's really important, and I'm hoping that we'll be able to overcome all these obstacles to eventually bring that to patients.
Okay, I've asked a lot of them. I'll open out for one or two. I know we're just over time, but I'll open up if there's one or two pressing questions that I missed. That's absolutely key if anyone wants to unmute themselves. If not, we're going to be scheduling some meetings with Olivier post his talk to discuss our organoid programs and others. If anyone's interested in attending those meetings, they can always email me. They can sit in on our meetings, or I'm sure Olivier, speaking on behalf of him, he'll be happy to chat with people.
Absolutely, yeah, absolutely. Really, I think that's as I said at the beginning, I think I would love to explore collaborations with you guys. Clearly a lot of amazing questions. Really, I'm actually very impressed with the quality of the questions. Actually, when I give this talk, here I'm not getting the same quality. So this is very promising. Clearly, there's a lot of people interested in organoids, at your IPM, and so I would love to continue the discussion and hopefully figure out ways to work together.
Yeah, fantastic. So Olivier, thanks again for taking the time to present.
My pleasure.
And have a good evening.
Thank you so much for having me. Thank you. Bye, guys. Bye, everyone. Bye.